This diagram shows a cell undergoing meiosis. That's the process by which sexually reproducing eukaryotes, humans, other animals, plants, create sex cells that they pass on to their offspring. Meiosis is an essential AP biology topic, and this video is going to teach you what you need to know about it. But this video is about so much more. That's because meiosis explains how you got to be who you are. It explains why you're similar to your parents and your siblings, but also different. And that's because meiosis is the source of variation in sexually reproducing organisms. That variation comes about through two processes that we'll learn about in this video. One is called independent assortment, which is how you received a different array of chromosomes from your parents than your siblings did. And the second process is crossing over and genetic recombination. That's because during meiosis, what happens is that parents combine the DNA that they inherited from their parents, passing on to you brand new, never before existing DNA. First time in the universe, and that's the DNA that you have. Now, add to that the fact that during fertilization, the genes from two different organisms are combined. Unless you're an identical twin, there's never been an organism in this universe that has your exact array of genes. So let's take your awesome uniqueness and let's learn about meiosis. Hey, my name is Mr. W. I'm a singing retired AP biology teacher, and my mission is to help you learn biology. I've spent hundreds of hours creating lessons like this one that are optimized to help you learn about various topics in AP biology. I'm also the creator of the website learn-biology.com and of the Biomania AP Bio app. I'll tell you more about both of those later. We're going to start with this incredibly cute photograph because it illustrates two things. First, how meiosis and fertilization are how genes get transmitted from one generation to the next. It also shows how meiosis is an engine for creating variation, both between parents and offspring and among the offspring. So let's walk through the life cycle of sexually reproducing organisms as it relates to meiosis. In humans and other animals, we have mothers and we have fathers. In mothers and fathers, we have specialized tissue that can produce sex cells. In females, those are ovaries. In males, those are testes. Those cells, as we'll see in a lot of detail later on in the video, go through the process of meiosis. And at the end of the process, we have sperm cells and egg cells. The sperm fertilizes the egg, and that produces a fertilized egg, also known as a zygote. That zygote undergoes cell division and subsequent development where the cells differentiate until what you have is not an undifferentiated zygote, but you have a differentiated, sexually mature adult organism. Based on the life cycle of sexually reproducing organisms, we can divide cells into two types based on the number of chromosomes that they have. Some cells are diploid, and what that means is that they have two sets of chromosomes. That's represented as 2n, n referring to one set of chromosomes, 2n referring to 2. Now, all body cells, including the testes and ovaries, have cells with a diploid chromosome number. And what that means, again, is two sets of chromosomes. Why two sets? Because you inherit one from your mom through the egg, another through the sperm from your dad. And what that means is that each of these chromosomes are not identical. The word that we use to refer to them is homologous. They have similar information, but the information isn't exactly the same. They have similar genes, but the alleles might be different. The genes might have alternative forms. Now, in the sex cells, the egg cell and the sperm cell, the chromosomes are haploid. And what that means is they have only a single set of chromosomes. So diploid cells, haploid cells. Two sets of chromosomes, one set of chromosomes. There are additional terms for describing the different cells that show up during the life cycle of sexually reproducing organisms. So the cells in the testes and the ovaries that are diploid but undergo meiosis are referred to as germ cells. Those germ cells go through meiosis, and what results are gametes. Gametes is just a way to say sperm and egg collectively. So sperm cells and egg cells, or gametes, they're haploid as opposed to the germ cells, which are diploid. Now, the sperm fertilizes the egg, and that produces a zygote, a fertilized egg. And that cell divides and differentiates to become the various tissues of the body. And we refer to those tissues as somatic cells. And those are, like the germ cells, diploid. We're about to get into the details of meiosis. And before we do, I want to emphasize that familiarity with mitosis and the stages of mitosis is really useful. It's a prerequisite for understanding what happens during meiosis. So I want to review a few things. And I also want to let you know that I have a song, a mitosis rap, that'll walk you through all of these stages of mitosis, and the link is below. So to begin with, I want to emphasize that the process begins with an interphase. An interphase involves replication of DNA. As a result of that, by the end of interphase, as we move into prophase, each chromosome is doubled into two identical pieces that are called sister chromatids. Now, those chromosomes have condensed during prophase. They start to get manipulated by the machinery of chromosome separation, which involves the spindle and the spindle apparatus. And by the end of prophase, we get to metaphase, where the chromosomes are aligned in the middle. Metaphase begins with M. They're in the middle, also begins with M. Now, following metaphase, what happens is the spindle pulls those sister chromatids apart. Anaphase begins with A. Apart also begins with A. That's how you can remember that. And then we have telophase, where a nucleus reforms, and then finally they're pulled apart during cytokinesis. Having reviewed mitosis, we can now take a big picture look at meiosis. And the first thing I want to say is that meiosis is sometimes referred to as reduction division. Why? Because it reduces the chromosome number from diploid to haploid. We humans have 46 chromosomes in our germ cells, our somatic cells. After meiosis, the gametes will have 23 chromosomes, half the number that they started with as diploid cells. So, what's the big picture? As we indicated, meiosis and mitosis both start with DNA replication. And at the end, what you have is doubled chromosomes that consist of two sister chromatids. So that's the first thing that happens. The second thing that happens is referred to as meiosis 1, because there are two divisions that happen during the course of meiosis. So what that first division, meiosis 1, does is it separates the homologous pairs, those matched chromosomes that you inherited from your mother and from your father. Now, as a result, we have cells that are haploid, but the chromosomes are still doubled. Why are they haploid? Because there's only one set, one representative of each chromosome, but they're still doubled, and that's why we need meiosis 2. That's the second cell division, and that cell division results in pulling apart the sister chromatids, and what we get is 4 haploid gametes. Depending if they're coming from a male or a female, they'll either be sperm or egg, and there's some additional complications, but this is the big picture, and it's very important to have in memory the information on this slide, because all of this stuff is good material to be tested on, both in your class and on the AP exam. Now we can start talking about how meiosis actually acts as an engine of diversity creation. And it does that through two ways, and the first is independent assortment. Before we look at the details, let's just re-emphasize something. Those homologous chromosomes that you inherited from your mother and your father, they're not identical. So let's take a look at, for example, chromosome 1. Chromosome 1 is, like all the chromosomes, an array of genes. Genes are instructions for producing proteins. Now, your mom and your dad, they're not genetically identical, and the chromosomes that they passed on to you are not identical either. They might have genes that are the same, but those genes also might be different. So let's just look at chromosome 1. This is representing the maternal one, and this is representing the paternal one. So what we have here is that the first gene on this chromosome is being represented as being the same. You can think of genes as kind of recipes, and these homologous chromosomes are weirdly recipe books that have the same recipe on the same page, but the actual recipe might be different. So like, for example, this might be a recipe for tomato sauce, and it's the same in your mother and in your father. That's why they're both represented by a capital A. But this over here might be a recipe for making a certain kind of salad, and in this kind of salad there might be more peppers, and in this kind of salad there might be more cucumbers. So they're both recipes for a salad, but the way that that recipe actually gets implemented is slightly different. So that's what homologous really means. Those chromosomes are not identical, and that's going to be really important in understanding how meiosis generates variety. So the first way that meiosis generates variety is through this process called independent assortment. Let's see what happens. So we have interphase, and then during interphase, what happens is that the DNA is doubled, and at the end of interphase, we move into prophase. This is prophase 1. It's the prophase associated with meiosis 1, that first cell division, where the homologous pairs are pulled apart. Now, before those homologous pairs are pulled apart, they find each other. And in a way, this is quite poignant, because basically what happens is that in the body of a parent, let's say of the father, the chromosomes that the father inherited from his parents, your grandparents, well, those chromosomes find each other. So chromosome 1 from your mom, from the grandfather, finds chromosome 1 from the grandmother, and they come together and they embrace. And that's what you see over here. Now, for simplification, for understanding this, I've created a cell that only has four chromosomes total, 2 homologous pairs. And here you see the first homologous pair embracing, and here you see the second one. Now, during metaphase, the chromosomes go to the middle. And note that when they go to the middle, they can go to the middle in essentially 2 ways. You could play around with more combinations, but the ultimate result will be the same, and I'll show you that on the next slide. So here we see that the chromosomes have been brought to the middle so that we have paternal chromosome number 1 on the left, maternal chromosome number 1 on the right, in relationship to a cell equator that's running from the top to the bottom. And these chromosomes are being manipulated by the spindle in the same way as happens during mitosis. And for the second chromosome pair, the paternal one's on the left and the maternal one's on the right. So when we pull those apart during anaphase, remember anaphase, A for apartness, in this gamete we'll have this array of chromosomes. It'll be paternal chromosome 1 and paternal chromosome 2. In this one over here, since these are going that way, we have maternal chromosome 1 and maternal chromosome 2. But these chromosomes can be flipped relative to one another. So in this other germ cell, we have the maternal chromosome on the left, maternal chromosome number 2 on the left, and paternal chromosome number 2 on the left, and the first ones are oriented in the same way. Now again, you can create models of this and create other orientations, but what you'll find is that you can't create more than 4 different arrays of chromosomes in the gametes. And here in gamete number 3, we have paternal chromosome number 1, maternal chromosome number 2. In gamete number 4, maternal chromosome number 1, paternal chromosome number 2. And again, you can move these around, but you're only going to find 4 possibilities. And with 2 homologous pairs, 4 gametes are possible. And basically the math behind that is that, well, in each homologous pair you have 2 chromosomes. It's a pair. And in this case, it's 2 to the number of homologous pairs. So we only have 2 homologous pairs here, so it's 2 squared. Now, let's play with the math a little bit. If you had 3 homologous pairs, then it would be 8 gametes possible. So it's 2 to the number of homologous pairs because it's 2 cubed, right? 3 homologous pairs. And if you had 23 homologous pairs, as in humans, then the number of different chromosomal combinations in the gametes that you can produce is 8,388,608. That's 2 to the 23rd. So you look at your siblings and you're like, well, why aren't I exactly like you? It's because the math of this is pretty phenomenal. Well, what's the probability you'd inherit the same egg? Well, that's 1 over 2 to the 23rd. what's the possibility you inherit the same sperm? It's also 1 over 2 to the 23rd. Because those are independent events, you'd have to multiply them together. So it's 1 times 2 to the 23rd multiplied by 1 times 2 to the 23rd is 1 in 70 trillion. So that's one reason why meiosis is this phenomenally cool generator of diversity. As we've seen, independent assortment is an incredibly powerful engine for creating diversity during meiosis, but there's much more. There's another process that's called crossing over, and that's combined with genetic recombination. Here's what it's all about. So we talked about how during interphase, the homologous pairs line up and they embrace. Well, they don't just embrace. They actually have crossing over points that are called chiasmas. This coming together is called synapsis. And during synapsis, these homologous pairs actually exchange pieces of DNA with one another. You know, here they come together. So imagine my left hand jumping over and switching sides with my right hand. Kind of a grisly image, but that's really what it's all about. So at the end of the process, you had a maternal chromosome and a paternal chromosome, but what they wind up doing is they wind up exchanging little pieces of one another. So by the end, what we have is recombinant DNA. We have brand new chromosomes that you get to inherit that have never existed before in the history of the universe. And there's a couple of terms. When these chromosomes are together, there's 4 sister chromatids amongst the two homologs. So that's called a tetrad. So that's what the tetrad looks like before crossing over. And then afterwards, you have these recombinant chromosomes. Holy cow, so much diversity, so much uniqueness, and that uniqueness is in you. So meiosis creates genetic diversity in 2 ways. We've seen independent assortment, this random assortment of parental chromosomes from each homologous pair into the gametes, and then on top of that you have genetic recombination. So again, look at your siblings. Why are you different from them? There was no chance, unless in the case of identical twins, that you would have the same array of chromosomes and that those chromosomes would be the same. You're well on your way towards understanding everything that you need to know about meiosis to crush it on your next test or on the AP Bio exam. And what I'm about to do is walk you through the entire process step by step. Before we go there, I'd like to ask you to do a couple of things. First of all, please like this video. Second of all, please subscribe to this channel, Science Music Videos, where you will find all of this great material that's really going to help you master your biology course. I've got lectures like this. I've got many songs that are pretty unique in terms of biology education. I also want to really encourage you to leave me a comment, tell me how this video was in terms of teaching you about this material. And if you ask me a question, I promise that I will respond, and maybe I'll even make a video and dedicate it to you. I also want to tell you about 2 things that are really going to help you learn biology, and that's why you're here. The first thing is my website, learn-biology.com, and I want to take you over to that website. And here we are at the tutorial that aligns with this video. So this teaches about how meiosis creates variation. And then we get to a quiz. So you just click, and what would you have to do? Key idea behind independent assortment is that each pair of homologous chromosomes is divided up. What would be that? Independently. Now, if you didn't know the answer, you'd get a hint and you could answer that question, ultimately successfully. During metaphase 1, shown below, the way that maternal and paternal chromosomes line up is as, what would that be? As a coin toss. It's as, can you think about the rest? Are random as a coin toss. So we've got many questions here, 6 questions in that set, and then there's an explanation of crossing over, and then there's a quiz about meiosis and variation, and here's what the quiz looks like. As shown in the diagram below, the pairing up of homologous chromosomes is also known as, oh, we learned about that. That is synapsis. So I'll show you one where you get it wrong. In the diagram below, these paired homologous chromosomes can also be referred to as. So if you get it wrong, you get a hint about what the answer is, and then that question will go to the bottom of the set. So when you finish this quiz, you will know this material. So that's what learn-biology.com is all about. It's your pathway towards achieving incredible success in AP Biology. In addition to learn-biology.com, I also have an app. It's called Biomania. It has a subset of the material that is on learn-biology.com, but you've got flashcards, you've got quizzes, you've got interactive diagrams, literally in your pocket. You can study at any time, and it'll give you the support, the practice that you need to really learn biology, because you know learning AP Biology is not easy. There's a mountain of material. You need practice. You need to study in the right way. Learn-biology.com and the Biomania AP Bio app will get you there. We have a guarantee. There are links below. Check it out. Thank you. So now that we've talked about how meiosis is this engine of variation, we can walk through the entire process, and we'll begin with interphase 1. Interphase 1 in meiosis, there's a replication of the DNA. So by the time interphase 1 is done and we've moved into prophase 1, we have duplicated chromosomes consisting of sister chromatids. These are members of homologous pairs, and the homologous pairs find one another, and during a process called synapsis, they embrace and actually exchange bits of DNA, creating recombinant chromosomes. So that's one powerful generator of diversity. In metaphase 1, the homologous pairs are brought to the center of the cell equator, and then they're pulled apart during anaphase 1. And again, remember that the orientation of those homologous pairs is set randomly. Here we show both paternal chromosomes on the bottom and the maternal chromosomes on the top, but they could be flipped relative to one another. So that leads to independent assortment, which generates more diversity. Notice also, if you look really closely, each of these chromosomes is already a recombinant chromosome. It's already exchanged a little bit of DNA with one another. So we have both things in play. Anaphase 1, the homologous pairs are pulled apart. In telophase 1, a new nucleus will reform. We have haploid cells, but they're still with doubled chromosomes consisting of 2 sister chromatids. Now, following that, there'll be a cytokinesis. There'll be an interphase 2 that's not shown in this diagram. An important point is that during the second interphase, there's not another duplication of the DNA. We then move into prophase 2, where the chromosomes condense again. These chromosomes are recombinant chromosomes because of stuff that happened during meiosis 1. They get pulled to the middle of the cell, and then during anaphase 2, the sister chromatids are pulled apart. And when you think about it, this is yet another opportunity for generating diversity, because the sister chromatids can be oriented one way or the other way, so there's yet more combinations that happen. Not quite as important as what happens during meiosis 1, but nevertheless is something to be aware of. Then we move into telophase 2 when the nucleus reforms, and then finally at the end we have 4 haploid gametes. I got a really fun song about meiosis, so won't you give it a try? You won't believe how much you'll learn about biology.